Welcome dear participants, welcome to the SOYAM course. This is the week 6 lecture number 5 and module is endocrine system, and in this we will today discuss about the hormones of the pancreas, pineal gland, thymus gland and the gastrointestinal tract. So first we will discuss means hormones of pancreas. As we know that pancreas is act as a both endocrine as well as exocrine gland. Exocrine part is the pancreatic enzymes, and the endocrine part is that different cells are there in the pancreas, that is called as islets of Langerhans, where the different types of cells are there that secrete different types of hormone. So exocrine pancreas coming in through the pancreatic acini, that these follicles are there that called as a pancreatic acini that secrete their cells through the pancreatic duct. So that is the pancreatic enzyme. So that is the exocrine part of the pancreas, and in between the exocrine part of pancreas, you can see that there is a group of cells called as islets of Langerhans, and this group of cells, different cells are there that secrete different kinds of hormones, and these islets of Langerhans are surrounded by the highly vascular tissue. Vascular blood vessels are there that provide the blood circulation to the group of cells. So basically there are 4 types of cells are there in the islets of Langerhans. That cells are the alpha, beta, delta and pancreatic polypeptide cells. So if we will talk about the different type of cells, then this beta cells, which is around 60 to 70% of the total cells of the islets of Langerhans, that secrete the important hormones that is called as insulin. Then comes to the alpha cells that correspond to around 20% of the total cells, and it secretes the important hormone that is called as glucagon. Then delta cells are there that correspond to around 5%, and that secrete the important hormone called as somatostatin, and around 5% is the pancreatic polypeptide secreting cells that have mainly the inhibitory actions. So if you see the insulin, how they are made up of different means a number of amino acids. And this insulin means if you talk about the human insulin, it is made up of total 51 amino acids, and in this 50 amino acids there are 2 important chain are there, A chain and B chain. A chain contains 21 amino acid and B chain contains 30 amino acids, and this A chain and B chain are connected by 3 disulfide bond. So in insulin, that is made up of 51 amino acid that have 2 chain, A chain and B chain, and both the chains are connected by 3 disulfide bond. So 3 disulfide bond are present in between these 2 chains. So if you talk about the synthesis of insulin will also takes place in the same manner as we have discussed for the synthesis of protein hormone. Here also first there will be the formation of the pre-proinsulin, and this pre-proinsulin have 2 important terminal, that is the N terminal and the C terminal, and this N terminal have one signal peptide. So this formation will takes place in the endoplasmic reticulum, and then this pre-proinsulin will be cleaved into another insulin that is called as a proinsulin, where the signal peptide will be removed and only the A chain, B chain, and there will be the connecting C chain. So this will present in the proinsulin. Again this proinsulin will be cleaved by the enzymes to formation of final form of insulin, where the C chain, which is the connecting chain, will be removed. C chain will be removed, and then the A chain and B chain will remain. So the active form of insulin have A chain and B chain, and C chain will be removed. So that is and then this equimolar concentration of insulin that contains the A chain and B chain and C chain they will packed in a Golgi vesicle and then remain stored, and whenever requirement will be there, they will be used for the another actions. Now, what is the role of insulin for the glucose transport? As we know that insulin secretion will take place during the hyperglycemic conditions, means when the blood glucose concentration increases in the blood, at that time the insulin secretion will take place. So the insulin promotes the uptake of the glucose from the blood by insulin dependent tissue such as the muscle and the adipose tissue. So it will help in the uptake of glucose. So whatever the glucose is there, whatever the increased glucose concentration is there in the blood, that will be taken by the insulin. So it will enhance the glucose utilization by the cells, so that the cells will utilize the glucose, and then whatever extracellular concentration of glucose will be there, that will be minimized. So this will be done by important type of transporter is there, that is called as a glucose transporter 4, GLUT4. So this transport will takes place by important transporter that is called as a glucose transporter, and then this glucose transporter will take the glucose into the cells, and once through the exocytosis this glucose will enter into the cells with the help of important mechanism that is called as a tyrosine kinase, and with the help of this tyrosine kinase this glucose will enter into the cells. Once the hyperglycemic condition will be removed, then this mechanism will be vanished. Now what is the insulin and its metabolic effect? So as we know that glucose concentration is in most of the mammals around 80 to 90 milligram per deciliter, but in case of the ruminant this glucose concentration is around only 55 to 65 milligram per deciliter. Why this glucose concentration is less in ruminants? Because the end product of carbohydrate digestion is not glucose, but instead of glucose it is a volatile fatty acid. So that's why the glucose concentration in ruminants is little less compared to that of other mammals. The important discovery for the insulin has been done by the Banting and Best in 1922, and for this discovery they got the Nobel Prize. So basically the insulin will takes place whenever hyperglycemic condition will develop. Hyperglycemic condition means when the blood glucose concentration will increase in the body, at that time this insulin concentration will increase. And what insulin will do? It will causes the peripheral utilization of blood glucose. So whatever the glucose will be there, their utilization to the cells will be done by this insulin. So nearly all cells have the insulin receptor, but important exception is there, that brain, erythrocyte, mammary gland, renal cortex cells and intestinal epithelial cells, they does not require the insulin in order to take the glucose from the blood. So these cells are independent of the insulin for the glucose utilization, but other cells require the insulin. Other cells require insulin means especially the muscle cells and the adipose cells require the insulin for the glucose utilizations. Also insulin promote the triglyceride synthesis in the adipose tissue and the glycogen in the muscle and the liver and the protein reserve in the muscles. So in this way that's why insulin is called as a anabolic hormone. Anabolic hormone means they will enhance the triglyceride synthesis, they will enhance this glycogen synthesis in the muscles and also help in the development of the protein reserve in the muscles. So that's why insulin is called as a anabolic hormone. So what is the effect of insulin on the carbohydrate, protein and the fat metabolism? So insulin causes the rapid uptake, storage and use of glucose by almost all tissue of the body, especially the muscle, adipose tissue and the liver. So in the liver, what it do? Insulin depress the rate of gluconeogenesis. Insulin decrease the rate of gluconeogenesis in the liver. It will promote the protein synthesis and the storage, and also it will promote the fat synthesis and storage. So it will depress the gluconeogenesis, it will enhance the protein synthesis, and it will promote the fat synthesis and storage. So that is the important functions or important role of insulin. So related to insulin, one important disease there that is called as a diabetes mellitus. So diabetes mellitus is defined. Diabetes mellitus is called as a syndrome of impaired carbohydrate, fat and protein metabolism that is caused by either lack of insulin secretion or decreased sensitivity of tissue to the insulin. So this diabetes mellitus may develop either due to lack of insulin secretion, means if insulin secretion will not be proper, then it may develop, or it may develop when the cell will be insensitive to the insulin, means the proper amount of insulin secretion will take place, but cells will unable to utilize the glucose or they will unable to use that insulin. So insulin resistance is developing in the cells. At that time also this diabetes mellitus condition is developing. So this is diabetes mellitus is mainly lifestyle diseases and it occurs mostly in case of the human, dogs and cats. So these are the species where this diabetes mellitus conditions are developing. So these type of animal are characterized by high blood glucose concentration. These animals species or the human being have the high blood glucose concentration because the insulin either it is not producing, or if it is producing, it is unable to utilize the glucose into the cells. So that's why the hyperglycemic like condition, high blood glucose concentration are present in the blood. So there are generally two types of diabetes mellitus is there. One is called as a type 1 or also called as a juvenile diabetes, and is also called as a IDDM, means insulin dependent diabetes mellitus, that is caused by the lack of the insulin secretion. So that is the type 1 or juvenile. Second is called as a type 2 or adult onset diabetes mellitus, also called as a non-insulin dependent diabetes mellitus, NIDDM, which is caused by the decreased sensitivity, means it may develop due to the insulin resistance to the target tissue. So here the resistance will be developed against the insulin, so insulin will be unable to use the glucose into the cells. So that is the two type is there, type 1 and type 2, and both the types what is happening, there is a higher level of blood glucose in the body. Because why higher level is there? Because body cells are unable to utilize the glucose. So first we will discuss in brief what is the type 1 or juvenile diabetes. This occurs because the beta cells are getting destroyed. Beta cells are getting destroyed, so here the insulin production is lost. And this type of diabetes occurs in the younger age of animals and the human beings. So it is maybe it is said as it is a genetic type of disease that means carry over from one generation to the another generations, and it may occur due to the viral infection, or it may due to the autoimmune disorders that involves the destruction of the beta cells. So once the beta cell will be destroyed, naturally the insulin production will be lost, and if insulin production will be lost, the glucose utilization to the cells will not take place, and then there will be the higher occurrence of glucose in the blood. Next is the type 2 or adult onset diabetes. It is most common type of diabetes mellitus. In this, the beta cells will produce the insulin, however the tissue will fail to respond to insulin. Here the proper amount of insulin is producing, but tissue will be unable to respond to the insulin. Insulin does not have any role in utilizing the glucose to the cells. So why it is happening? It is happening due to the development of resistance, or it happening due to the decline in the receptors for the insulin. So that's why this type of diabetes are occurring, and mostly this type of diabetes is associated with the obesity. If obesity is there either in the animal or the human being, that they are most prone to the development of the type 2 diabetes mellitus. So what is called as insulin resistance? So insulin resistance is primarily cause of the type 2 diabetes mellitus. It is a condition by which the insulin induced glucose uptake is reduced in the insulin sensitive tissue. So insulin induced glucose which are normally occurring in the tissue, that type of condition is not developing, because that means tissue is not responding to the insulin, so they are unable to utilize the glucose, and it occurs due to the inhibition of the insulin signaling pathway. So insulin signaling pathway is not functioning properly, that's why this type of condition is developing. So it is believed that insulin resistance lead to the hyperinsulinemia. Hyperinsulinemia means there is a higher secretion of the insulin when the beta cells produce large amount of insulin in an effort to control the blood glucose. So since there is a hyperglycemic conditions, so body produce insulin to combat that increased blood glucose level, but the insulin does not have any role in utilizing that glucose. So that's why in the body the insulin amount is higher. So that's why this condition, that is the hyperinsulinemia, is developing. So there are certain differences there in the type 1 and type 2 diabetes mellitus, is that in type 1 diabetes there will be the hyperglycemia, quite natural is there, blood glucose level will be higher, but there will be the hypoinsulinemia, means insulin production will not be proper. So hypoinsulinemia is there, but hyperglycemia. So hypoinsulinemia and hyperglycemia, but in case of the type 2 diabetes mellitus, there will be the hyperglycemia as well as the hyperinsulinemia. So both glucose concentration as well as insulin concentration will be higher in the type 2, whereas in type 1 there will be the hyperglycemia, means increased glucose concentration, but the insulin secretion will be less. So, what are the symptoms of diabetes mellitus? Naturally, the blood glucose will be very high. There will be the loss of glucose in the urine. There will be the dehydration. There will be polyuria and increased thirst. More urine formation will take place, and if the more urine will be lost from the body, the animal or person will be feel for thirsty. So, increased thirst will be there. Then there will be the depletion of body protein. Since the glucose is not available, so the other metabolite like protein and fat will be utilized. So, there will be the depletion of body proteins, and the chronic high glucose concentration causes the tissue injury, increased risk of the heart attack. There will be a stroke, or in the stage the kidney disease. There will be the retinopathy. There will be the blindness and the gangrenes of the limbs. So, these are the very important symptoms of the diabetes mellitus. Blindness may occur, or end stage kidney disease may occur, or if there's any kind of wound is there, that wound will not heal at a proper time. So, that is the important symptoms of the diabetes mellitus. Now, what is the physiology to diagnose diabetes mellitus? Diabetes mellitus, how you can diagnose? One important test is there, that is called as a glucose tolerance test. So, in this test, what actually is done, animal have to give some amount of glucose to ingest. So, in normal person, if they will ingest some amount of glucose, their glucose concentration initially it will increase, but it will come to down level by after 1 to 2 hours. But in case of a diabetic person, if they will ingest some kind of glucose externally, their glucose level will increase to much higher level, and they will not come to the normal level. So, that is the important test to diagnose any kind of diabetes mellitus, that is called as a glucose tolerance test. So, in this way we can test the whether the person is having the diabetes mellitus or not. Now, what is the treatment for the diabetes mellitus? Treatment is that there should be the exogenous administration of insulin. Since the existing insulins are not working properly, so we may give the insulin from the outside. Then another thing is that the person or the animals, if the overweight is there, they should lose their weight. So, if the weight will be lost, then the insulin will be responsive to the tissue, and then that can be corrected. Then we have to monitor the glucose concentration every day, so that we can give the effective treatment. And then administration of insulin after each meal. After each meal we have to give the insulin injection, so that it can maintain the blood glucose concentration. That is the hormones, the insulin which is secreted from the beta cells. Another hormone is called as a glucagon, which is secreted from the alpha cells of the islets of Langerhans, and these have the opposite action to that of insulin. Whatever the action is there for the insulin, the glucagon have the exactly opposite to that one. It means what it will do, it will stimulate the glycogenolysis, causing the release of glucose into the blood. It promotes the gluconeogenesis. That was the not case for the insulin. And glucagon activate the adipose cell lipase, making the increased quantity of fatty acid available to the energy system of the body. So, this action is quite opposite to that of the insulin. Another hormone is called as somatostatin, which is a peptide hormone secreted from the delta cells of the pancreas, and it is also secreted by the enteroendocrine cells scattered throughout the intestinal tract. So, somatostatin also produced by the hypothalamic neurons, where it often have a growth hormone inhibiting hormone. So, it have a mainly inhibitory actions. It will inhibit the production of both insulin as well as the glucagon. So, this somatostatin have inhibitory functions, and it will inhibit the production of both insulin and the glucagon. Another hormone is called as a pancreatic polypeptide. It also have a inhibitory actions. It will also inhibit the action of the both insulin and the glucagon, and it will pancreatic polypeptide inhibit the pancreatic exocrine secretion. It will also inhibit the gallbladder contraction, also the gut motility. Basically, it have a very inhibitory actions. So, that are the hormones which is secreted from the pancreas. Now, we will see next gland is the pineal gland. So, pineal gland is a small collection of cells arising in the center of the brain in a groove where two halves of the thalamus comes together. So, it is present in the brain, and this pineal gland is referred as a third eye because its activity is influenced by the amount of light the animal is exposed to. So that's why it's called as a third eye, because it's very sensitive to the light. The pineal gland is also referred as a epiphysis cerebri. The main hormone which is secreted from the pineal gland is the melatonin, and this melatonin is synthesized from important amino acid that is called as a tryptophan. So from tryptophan, this important hormone, that melatonin, is getting synthesized. So in case of the mammals and the birds, the light and dark information is transmitted from retina of the eye to the pineal gland via the various nerve. So light and dark information is mainly perceived by this melatonin. Secretion of melatonin is stimulated by the darkness and inhibited by the light. So that's why we say during sleep time more melatonin synthesis and is taking place in compared to the daytime. In daytime more serotonin, another hormone is there that is called as serotonin. So in the daytime when we are wakeful, at that time more serotonin secretion is taking place, but when we are sleeping at the night or the dark, this more melatonin is secreting. So high level of serotonin increase the wakefulness, whereas the high level of melatonin will induce a sleep like state. This pineal gland is responsible for regulating the circadian rhythm that affect the sleep pattern. Circadian rhythm is means the activity or rhythm which is taking place in the 24-hour cycle. So this pineal gland is mostly regulating the circadian rhythm and also affect the sleep pattern, and it will determine the periodicity of estrous cycle in the female. So it's a very important hormone related to the reproduction also. In case of seasonal breeder, melatonin can increase or decrease the gonadal size and the development of in the male. Now we'll see how it will affect the gonadal activity in the seasonal breeders. So it will inhibit the gonadal activity in the long day breeder like hamster and the horse. Why it will inhibit the gonadal activity? Because long day breeder means there is a increased amount of light, duration of light will be more, so less melatonin synthesis will take place. So that's why this inhibit the gonadal activity, whereas it will enhance the breeding gonadal activity in the short day breeder like sheep. It will enhance the gonadal activity. Also it is said that birds are induced to lay egg around the year if the light and dark period is balanced properly. So nowadays means modern hatcheries are there, their light and dark adaptation are maintained properly, means how much amount of light should be provided and how much amount of darkness should be there. So if we can control this light and dark period for the hen, we can get the egg production around the year. So that is the main role of here the melatonin, how it is controlling the animal productions. Next gland is the thymus gland. This thymus gland is a lobulated lymphoid organ located in the thoracic cavity, midline within the chest, anterior to the heart and space between the lungs. This thymus gland is present, and this thymus gland is very developed up to the puberty, but as the age advances it will decrease in size and slowly it will be replaced by the fat. So what the important function of this thymus gland is that it helps in the maturation and proliferation of the T cells. So it have a very important role in the immunity. And what hormone this thymus gland is secreted? This thymus gland is secreted very important hormone that is called as a thymosin, and this thymosin will stimulate the maturation of T cells and involve in the cell mediated immunity. So if some individual with absent or aplastic thymus, they are susceptible to the recurrent infection because there is a underdeveloped thymus gland, so they will be very recurrent to the infections. If thymus gland is not there, they will be very susceptible to the infections. Next hormone is the called as a gastrointestinal hormones. So these are the hormones which are secreted from the gastrointestinal tract. There the different cells are there in the gastrointestinal tract that secrete these hormones. So what are the hormones are there? It is these hormones are getting synthesized. How synthesis and release of gastric hormone is taking place? Gastric hormones are synthesized in specialized cells of the GI tract mucosa known as the enteroendocrine cells. These enteroendocrine cells are like stem cells which are regenerating itself and these enteroendocrine cells are mainly responsible for the secretion of gastrointestinal hormones, and these are dispersed throughout the gastrointestinal mucosa. So now we'll see what are the important hormones are there that secreted from the gastrointestinal tract. Important hormones are there: gastrin, secretin, cholecystokinin, GIP, then motilin, somatostatin, and incretins. So we'll see in brief what are the important functions of these hormones. So gastrin, mainly it will stimulate the hydrochloric production and also pepsinogen secretion, and it will enhance the gastric motility. Very few hormones are there in the GI tract that will enhance the gastric motility, and one of the hormone is the gastrin. Next hormone is secretin. It will stimulate the sodium bicarbonate production, secretion from the pancreas. It will inhibit the GI motility. Then cholecystokinin, it will stimulate the pancreatic acinar cells to secrete pancreatic enzyme, and it will also inhibit the gastric motility. Then GIP, it will inhibit the gastric motility and secretion similar to that of CCK, and it will induce the satiety, means the fullness. It also called as a fullness hormone. Then motilin is a very important hormone. It increases the gastric motility by stimulating the MMC, migrating myoelectric complex, and it will prepare the gastrointestinal tract for the next meal. So whatever the leftover is there, that will be removed by this motilin hormone because it causes the increased gastric motility. Somatostatin, as we told, that it is inhibitory in nature, and then incretin also decrease the gastric emptying, and it will induce the satiety. So these are the important hormones which are secreted from the gastrointestinal tract. Then another last one is called as a pheromones. Pheromones are a chemical signals that release into the environment by a gland and travel through the air or water to the sensory cells in another animal. So pheromones secreted from one animals, it comes into the airs, then going into the other animals to do their actions. So that signals used for the sexual activity, such as the signaling or the readiness to the mate, or it will help in the marking of the territories. That marking of the territory means dog used to urinate at some place so that they mark their territory, that this is their area, and other behaviors related to the interactions among the individuals of a species. So pheromones have a very important role in the signaling. Important for pheromones in which is the methyl para-hydroxybenzoate that released from the vaginal secretions, that means secreted during the pro-stress or the sexual receptivity time, and when the male dog will perceive that, they will go for the matings. Boar also produce the sexual attractant in their preputial pouch and also in the submaxillary salivary gland. So this female pheromones associated with the vaginal secretions and it comes in the urine during the stress cycle, and this will induce a phlegm response. Phlegm response is very peculiar features that occurs in a stallion and also in the bulls. In this case, what is happening? There will be the elevation of head elevation will take place, and the curling of the upper lip, that is best seen in the stallion and the bulls. So there will be the head elevation and the curling of the upper lip, and they will press the upper part of the mouth, and then they will perceive the whatever the pheromones are secreted that is coming in the urine, that will perceive. So important organ is there that perceive this pheromone secretion, that is called as a vomeronasal organ. So this vomeronasal organ is nothing but it is accessory olfactory glands that sense the release volatile substance that is present in the pheromones, that is perceived by this vomeronasal organs, and then the subsequent action will be done. So that is the important role pheromones play in the signaling. So that leads to the completion of this endocrine system. Today we have discussed about the this hormones of the pancreas, and then we have discussed about the pineal gland, the thymus gland, their hormones. Then we have seen the what is the gastrointestinal hormone, important hormone we have discussed, and at the end we have discussed about the pheromones. So in this way we have completed the this our modules, the endocrine system. Thank you.